So we're on plate 16, lecture 16. Plate 71, we're going to do a little change in the methodology. We've got an overhead camera on this, so bear with me as I kind of learn that. So we're going to start with organization and function of the digestive tract. And again, this picture is a great human picture. If you want to think of what a digestive tract in a Cocker Spaniel looks like, it looks kind of like that. There are a number of modifications that occur in our domestic animals that facilitate economy, that facilitate the ability to utilize other unusable substrates. But by and large, the overall scheme is similar. So we're going to use this as our model, and I'm going to start with a general overview of the digestive tract. And digestion begins in the oral cavity. The oral cavity, mouth, associated with the tongue, gums, teeth. And processing actually begins at this anteriormost portion. The food bolus is then passed from the oral cavity back into the pharynx, and we talked about the pharynx as being this common cavity between the respiratory tract and the digestive tract. So now we've kind of gone into the digestive end of it. On the dorsal surface of the pharynx sits the esophagus. That becomes important, particularly if you're passing endotracheal tubes. You want to make sure it goes down in the trachea, not into the esophagus. And if you want to do endoscopy of the stomach, looking for ulcers, you want to make sure that the tube passes in that direction and not down into the trachea. The esophagus feeds into the stomach. From the stomach, you move into the small intestines, and from the small intestines, you move into the large intestines. So we have what is referred to as this enteric pathway, or GI tract. And you can have problems anywhere along that tract. You can also have manipulation and addition of compounds. And so we're going to kind of walk through that. Digestive glands. Digestion begins in the oral cavity with saliva. It's continued and processed through the stomach and the components of the enteric system, and also the extraenteric, things that feed into the GI tract. The liver, the gallbladder, pancreas are all these digestive glands that sit outside the confines of the regular tract. In terms of function, digestion and breakdown occurs in the anteriormost region of the GI tract. It is the site of the intestines where there is primarily digestion and absorption. And then absorption and rehydration or dehydration occurs in the posterior or colon end of it. Enzymes transfer food into absorbable nutrients. So what do we need in this class? What do we need to know about nutrition? Not much, right? The three basic food substrates: proteins, carbohydrates, and lipids or fats. In the proteins, you have different size proteins. Some are long, some are short. In some cases, the proteins may only consist of amino acid units. The idea in digestion is to break those compounds into the most absorbable forms. So for proteins, that is amino acids. And you'll see the focus of digestion to break down proteins into the amino acids. Carbohydrates, similarly, they can be complex, like starch molecules. They can be single molecules, like glucose. They can be two glucose moieties stuck together, like lactose, so we talk about milk sugars. So the term sugar and carbohydrate and starch sometimes are used interchangeably, but recognize that we're looking at the simple sugars, monosaccharides, are the ones that we absorb. And so digestion needs to break down these complex and semi-complex carbohydrates into absorbable sources. For fats, Triglycerides, we talked about triglycerides as being the storage form for fats. Triglycerides must be broken down into absorbable subunits, which are the fatty acids. Now, a triglyceride consists of 3 fatty acid chains linked to one glycerol. Glycerol, in fact, is a carbohydrate component, but in the absorption process, what you want to do is you want to cleave off those fatty acid acyl groups so that they can then be absorbed. Absorption then occurs in the GI tract, very vascular, a lot of blood flow during the vegetative state, so under parasympathetic control, blood flow to the GI tract is increased, activity of the GI tract, movement of the muscles is increased to help both in passing the material down the GI tract and churning it up and mixing up in a digestive process. So let's start the process in the oral pharynx. Plate 72. Teeth structure, dentition is a huge thing in domestic animals. And in your different classes, you will touch on each of those elements and talking about the differences and the modifications that occur, particularly in browsers. But by and large, the embryological basis, the kind of the functional origin of these teeth are very similar. That even herbivores have incisors, and incisors are involved in cutting. Omnivores, carnivores, characterized by having a particularly long set of teeth, what are called the canines. In your notes, they talk about these as being involved in tearing. Canines are also great in holding the prey item. Very effective predators. If you've ever been snatched by a cat, it's those canines that go into you, those teeth. Primates, same thing. Something that scares me to death is looking at the length of some of those canines, which are very effective in capturing a prey item and holding the prey item while then you can go to work on it on your incisors. There are premolars and molars in humans. These are grinding surfaces, so there's tearing, cutting, and grinding. Note that also one modification would be this idea of these very rough ipsodont teeth. They're very good at grinding, and also things like carnassial. The carnassials in dogs. So when you're looking at a dog chewing on a bone, one of those first premolars has really been adapted into kind of helping tear the flesh, grind the flesh off the bone. So the idea is to reduce the size of the food particles. Now, you would never know that in my dog, who is able to consume entire boluses about the size of a small melon in one phenomenal gulp. But remember what Mom said, chew your food 50 times, or was it 150 times? Reduce the food particles. So that allows an increased surface area for the action of digestive enzymes. So we've started the digestive process with mechanical, and we have our salivary glands, parotid, mandibular, and sublingual glands. So there are a number of salivary glands which are exocrine glands. An exocrine gland is a gland of specialized tissue which produces a product that then gets dumped into a duct or tube and then is delivered to the site. An endocrine gland is one which produces a compound which is circulated through the blood. Exocrine versus endocrine. Saliva glands are exocrine glands, and here's an example of a serous and a mucus. And they talk about the acini, which are these little follicles or structures composed of these cells. In lab, you'll have the opportunity to look at some of these salivary glands in a bovine lip, where the cells are very predominant, very enlarged, because they produce large amounts of product, which are then transported across the membrane into this ductal tissue, and these salivary ducts then take this solution. You have saliva, which tends to be relatively serous or watery, and you also have the mucus-producing salivary glands. And it's this combination of this watery mucus that produces this wonderfully impressive bolus of macerated food, which aids in the transport down. Saliva contains water. It also contains enzymes. Your notes talk about 1.5 liters of saliva each day. Humans, small time compared to cattle. Huge, copious amounts of saliva. And they're using and recirculating a lot of that fluid, aiding in the digestive process in the rumen. Water environment. We talked about the importance of having a thin water line of the alveoli. So there was a water interface between a chemical and the body. We see the same thing in terms of olfaction and taste buds. In order for the chemical to stimulate a taste bud, it must be dissolved. It must be in an aqueous environment. So part of the serous component of saliva is meant to solubilize these taste particles and allow them access then into sensory taste buds located along the tongue. So that's an important component. The other component, another important component are enzymes. And the enzyme, the primary enzyme that is produced by these glands is ptyalin. Ptyalin. Sounds like you're spitting it, doesn't it? Ptyalin. Ptyalin is an example of an amylase. So amylases are enzymes that are involved in carbohydrate breakdown. One example of an amylase is ptyalin. So we start the digestive process in the mouth. Another enzyme component of saliva are lysozymes. We talked about I think you may have been queried on an exam about a lysozyme. Remember that lysozymes were intracellular organelles that packaged the lysosomes packaged the lysozymes, which are involved in the denaturation of viruses and bacteria. So these enzymes are bactericidal, microbial-cidal. The presence of these lysozymes helps combat the introduction. What a wonderful introduction into your body for microbes is by the mouth. Dirty hands, touch tables, goes to your lips, it goes to your food. It's a wonderful source for inoculating yourself with all kinds of disgusting bacteria and viruses. Lysozymes help protect against that. You look at a dog licking a wound, and your first thought is, oh, this dog just licked its butt, now it's licking a wound. Is this sanitary? Well, canines, dogs they spend a lot of their life eating material that may not be the best, right? Humans, much more sensitive about it. Canines, it's a dead pigeon. It's been out there a couple days. The dog goes out and eats it robustly, and yet doesn't seem to suffer the same maladies that you and I would get. High levels of lysozymes. Those are good things. So content and function of saliva. Water for dissolving the bolus for taste, lubrication for the bolus, enzymes, amylase, starch digestion, and the lysozymes that have an antibacterial action. You need to move the food particle back. So you have nerves, cranial nerves, that are involved in tongue motion, and the tongue then pushes this nice macerated bolus, wet, slippery because of the mucus, into the esophagus, and where you then initiate a peristaltic wave. So peristaltic waves are very characteristic of smooth muscle. We talked about, smooth muscle way back when, and its involvement in the bladder, its involvement in the GI tract. You didn't have to produce forceful contractions, but you needed to accommodate a large range of muscle sizes or muscle lengths. Exactly what's happening when you put that bolus of pizza back into the esophagus. The esophagus swells in size. You still want that smooth muscle to be able to contract and move it in a peristaltic or wave fashion down the esophageal tract. Some animals have lots of skeletal muscle up in the anterior portion of the esophagus. Humans are virtually all smooth muscle. So peristalsis propels the food bolus into the esophagus. Okay, plate 73. We're now into the stomach. The esophageal or cardiac sphincter, right here. Your notes have it as the esophageal sphincter. Sometimes it's also referred to as the cardiac sphincter. So this is esophageal tissue. There is and this is longitudinal muscle as well as circular, so that it's able to compress in this fashion and move the bolus in an anterior to posterior direction. The sphincter muscles are characterized by rings of smooth muscle at right angles to the longitudinal. So these then are the guardians or gateway between the esophagus and the stomach. It's this sphincter which contracts when you're upside down on a monkey bar to prevent fluid and GI contents from the stomach from going back. This is also the sphincter that becomes weak in gastric reflux disease. So in fact, where you actually will get movement of an acidic stomach contents back up into the esophagus. Very serious, very big concern, both in humans and in domestic animals. So these sphincters, there's one on the entrance into the stomach, a esophageal or cardiac sphincter, and then there is one at the pylorus or pyloric sphincter. That then defines, those two sphincters define the entrance and the exit for the stomach. There is the fundic, protracted and large anterior portion of it, the body, and then the final portion is the pyloric region. And this shows you a kind of a view, transverse section, that the walls of the stomach are waveform. They have an increased surface area. There tends to sometimes to be a glandular and a non-glandular region. But these are characterized by these what are termed gastric pits. So there are depressions then. This is showing you this depression. And these pits are lined with cells. And there are a number of cell types that comprise these gastric pits. The first major cell type are the parietal cells. Parietal cells are involved in the secretion of hydrochloric acid. The hallmark of the stomach is an intensely acidic environment. We talked about normal pH being 7.4. A pH of 3 is not uncommon in the stomach. And remember, these are log changes, so that's a several thousand-fold difference in terms of the hydrogen ion concentrations. Those hydrogen ions come from the parietal cells. So if we look at these cells, where is that hydrogen ion coming from? Where else but CO2? And what's going to make this all happen? Carbonic anhydrase. So take the picture. You've got carbonic anhydrase in red blood cells. That facilitates the movement of CO2 into the red blood cells at the tissue levels. You have carbonic anhydrase in the kidney cells. What are you doing? You are reabsorbing bicarbonate. You are adjusting pH balance. What are you doing in the parietal cells? You are trying to produce high levels of hydrogen ion. So CO2 is pulled into the parietal cell because of the presence of carbonic acid, or carbonic anhydrase. This enzyme catalyzes the combination of CO2 and water and produces a carbonic acid. The bicarbonate is moved out in this direction. The hydrogen ion is actively transported, so there's a proton pump. In fact, a number of pharmaceuticals are aimed at regulation of the action of this proton pump in terms of controlling and regulating excess acid secretion. So the presence of these pumps allows this hydrogen ion to be moved. Chloride moves passively in exchange for this bicarbonate. So there's a bicarbonate chloride link here to maintain neutrality. So this chloride then can combine with this hydrogen ion and get pumped in and produce these exceedingly high levels of hydrochloric acid. So you got a dachshund, comes into the clinic, vomiting. Oh yeah, sure, it's been vomiting all day, every day. Where's this animal going to be on an acid-base scale? It's losing acid from the stomach. We're vomiting from the anterior aspect, losing hydrochloric acid. This animal's compromised in terms of providing hydrochloric acid. So it produces more hydrochloric acid. That is going to produce more and more bicarbonate. So, individuals that present for sustained chronic vomiting are usually alkalotic. We talked about a respiratory alkalosis where you got rid of a lot of CO2. That's a respiratory alkalosis. Here's an example of a metabolic alkalosis, something that's driven by the metabolism of the animal. Hydrochloric acid, nasty stuff. How do you protect this? I mean, you're using it to digest things like proteins. Proteins that make up all the cells of the stomach. So how do you protect that? Another important component of the pit gland are mucus cells. And it's these cells that produce a protective polysaccharide covering that goes out, mucopolysaccharides, that protect and line the walls of these cells. So this hydrochloric acid tends to work on material inside the stomach, not on the walls themselves. The third cell type are the chief cells, or zymogen cell, kind of an old term. These cells are responsible for the production of an enzyme, pepsinogen. Angiotensinogen, you recall, was an inactive precursor. So that ogen suffix kind of lets you know that pepsinogen is an inactive enzyme. It requires the presence of hydrochloric acid to cleave or cut off a portion of it to make it active. Pepsinogen is the inactive form. Pepsin is the active form. It is pepsin which is the enzymatic agent to break down proteins into amino acids. Pepsinogen is sequestered within the chief cells. It does not auto-digest those cells because it's inactive. It requires the presence of that hydrochloric acid to make it active. And then the combined, the concert of the mucus protecting the cells, the acid to help in the digestion, and also the presence of this proteolytic enzyme. So carbohydrate metabolism starting in the with enzymes in the oral pharynx, protein digestion, enzymatic protein digestion starting in the stomach. But remember, all of this requires this kind of maceration. You're reducing the food particle size so that you have a greater surface area for these enzymes to work on. Gastric movement churn food to produce chyme. What a wonderful, you could make great lyrics with that. There once was a soldier from chyme. Chyme is the, if you don't hit clear, chyme is the product of food and hydrochloric acid. So once you kind of reach this point now, you've started in earnest the digestive process, you've created a bolus that is now referred to as chyme. And it is this chyme then that will continue or not continue its movement along the line of the GI tract. So now I think I can go to plate 74 and we can do this at I do need to talk about liquidity, gastric emptying and rate, because this has huge social implications for the upcoming weekend. The rate at which chyme is removed from the stomach is a function of its consistency. So a very liquid chyme, very digestible. So all you've been eating is beef bouillon. Not a lot to digest there. Not a lot of maceration involved. Hydrochloric acid is added in. It's incredibly liquid. It stimulates the sphincter muscles and increases gastric emptying. The presence of chyme, the presence of this acid environment, then further acts to decrease. So here's this chyme that moves into the first portion of the into the small intestine, called the duodenum. Cells in the wall of this tissue are sensitive to gastric secretions and will, in turn, close off the gastric sphincter. Pyloric sphincter. That's a way then of regulating and controlling the amount. Because in fact what you need to do initially is neutralize that acidic environment. Stomach acid, intestines not acid. We're going to develop a whole process to neutralize this acidic chyme. But in the presence of acid chyme, the movement is decreased. So the more of this tissue you have, the longer it takes to deacidify it, the slower the gastric emptying. So this shows you chyme in the chyme, a bolus, so there's a physical stretch. The next one emphasizes that it is also sensitive to hydrogen ion. So now while chyme has lots of hydrogen ion in it, these things go interchangeably. Similarly, if it's very liquid, it opens the sphincters. If this liquid is very acid and it goes in, the sphincter will then immediately close down, so that we can regulate the amount of material entering into the intestine and what is being presented for further digestion. The last example is the reason that you serve cheese whiz at cocktail parties. High lipid. Lipid is a very tough thing to digest, right? We haven't talked about lipid digestion yet. We've talked about carbohydrates, proteins, easier to convert those. Fats and water don't mix very well. And in high-fat diets, you want more time spent churning in the stomach to further decrease the size of the molecules for reabsorption, and that includes lipid. So a high lipid diet is going to restrict gastric emptying. So you're at a party, you're serving a nice wine. Some of your invitees are drinking too much wine. They have large amounts of alcohol in the stomach. we have virtually no absorption. Well, I should say, very limited absorption through the stomach, particularly compared to the intestine. So if you're able to slow the emptying of the stomach, you are going to slow the presentation of alcohol to the small intestine. So those are Martha Stewart tips based on the physiology of acid and chyme and knowing about the regulation of gastric emptying. All right, can we go to the, there we, excellent. Well, this is, I'll figure it out here one of these times. Or not. So, Bo, do you know how I get it on my screen here? No. So we're at what minute time? 30. Yeah, I need to go a little bit longer. That's way interesting. All right, so what I'll probably do is we'll try this. I can read it off this one. He says. You know, it's not one thing, it's another. Let's go ahead and just go to the overhead. That'll work for me. Hormonal regulation of digestion. We said that the gastric emptying is responsive to stretch and chemicals. And we, in fact, see exactly the same thing in the stomach, that it has stretch receptors. It has chemical receptors. Both stretching, entrance of food into the stomach, causing this kind of expansion, and the presence of these food particles stimulate the release of a hormone called gastrin. There are cells in addition to the pit, the gastric pits. There are cells on the wall of the stomach called gastrin secreting cells. This is a hormone secreting cell. So the response is that this cell is stimulated to release secretin. Secretin, as a hormone, exerts its influence through the circulatory system. But it's particularly interesting is that this cell is very close to the gastric pit cells, including the parietal cells, yet the mechanism of action is through a endocrine function. That is, this hormone is dumped into the blood, it circulates throughout the body, goes through the venous blood supply to the heart, to the arterial, and is delivered back to the stomach through an arterial supply in a concentration that is sufficient to stimulate that cell to release hydrochloric acid. So gastrin is very good at increasing the amount of hydrochloric acid secretion. Secretin is kind of the other way around. Gastrin is gastric, stomach, so it's involved in elevations of hydrochloric acid. Secretin is a hormone which is produced in response to an excess amount of hydrogen. You recall I said part of the mechanism, one of the components that regulates gastric emptying is the amount of volume of chyme and also the acid composition. That is modulated through these secretin-secreting cells, which are in the duodenum. So gastrin-secreting cells in the stomach, secretin-secreting cells in the duodenum, or the first part of the small intestine. And right there at the sphincter. So this is then the sphincter between the stomach and the small intestine. As acid chyme moves into the first part of the duodenum, high levels of acid stimulate the release of secretin. Secretin has two major roles. One is to inhibit hydrochloric acid secretion. So it's saying, I've got plenty of acid, you can tone it down. The second is that it goes to ductile cells of the pancreas. Now, not evident in this diagram is the pancreas, both an endocrine and an exocrine tissue located at the, physically it's located at the anterior aspect of the duodenum and the caudal border of the stomach. So you'll see pancreatic tissue right in here. We'll talk about the endocrine function of the pancreas later. Recognize that most of the cells of the pancreas serve an exocrine role. In large part, they are involved in the production of bicarbonate. It shows the exocrine cells dumping this bicarbonate into the ducts, so it's exocrine. The pancreatic duct feeds in immediately adjacent to these secretin cells and the presence of this high acid. This is the neutralization that you need to shut these things down. So, large bolus of chyme, very acid, slowing the emptying, waiting, all the while waiting for this bicarbonate to neutralize the acid. Two more hormones. We haven't really dealt with fat digestion. We've talked about how fat can delay gastric emptying. You're trying to get it into smaller molecules. We see the evidence of that in the duodenum. So in addition to secreting cells, there are cells which produce a hormone called cholecystokinin. Chole meaning bile, cysto meaning bladder, and kinin meaning to move. So this hormone stimulates the movement of the gallbladder. The gallbladder contains bile, important in emulsification in the digestion process, that gets dumped into, where am I? I'm over on, down here. So here are, these are cells of the pancreas as well, and the gallbladder. So the gallbladder is up here. Here's the pancreas. Gallbladder is dropping bile in. Cholecystokinin also stimulates the release of enzymes that begin the fat breakdown, that is the presence of lipase. So amylase starts in the oral cavity. We see peptidases or proteinases. The pepsin in the stomach. Now we've got our complete triad. We've got our lipase. It's being delivered into the duodenum. The last hormone of interest is a hormone released by cells, again, of the duodenum. This is gastric inhibitory peptide, GIP. Gastric inhibitory peptide reduces the action of the smooth muscle. It limits the amount of emptying, and it appears to be under the control of large boluses of chyme. So large masses stimulate the release of GIP, and also very fatty diets. Large amounts of lipid will produce an inhibition of gastric emptying, and the mechanism is through gastric inhibitory peptide. So I think we'll end there and continue next time.